microphone. He is the general manager of the Frontier Research Department at Amano Enzyme in Japan. Dr. Ishihara earned his PhD in biology at the University of Tokyo in 2005, carried out postdoctoral work in Japan, and joined Amano 15 years ago. He worked at Amano Enzyme USA in Chicago, or outside of Chicago, for 2 years and now leads the research in Japan on developing next-generation biocatalysts. Dr. Ishihara has made important contributions in enzyme innovation, including engineering protein glutaminase to improve the texture of plant-based foods. I think he's going to talk about that, and enhancing the enantioselectivity of Burkholderia cepacia lipase. I think he's also going to mention that. It's also a workhorse in fragrance and pharmaceutical synthesis. His team combines classical protein engineering with modern approaches, such as molecular dynamics simulations and machine learning guided design, pushing the boundaries of what enzymes can achieve in industry. And today he will speak on the performance expansion of industrial specialty enzymes, sharing how Amano is discovering, improving, and commercializing enzymes to more sustainable and precise industrial processes.
Thanks so much, Professor Romas Kazlauskas, organizing this symposium, and great introduction. So I'm Satoru Ishihara from Amano Enzyme. I'm honored to be here. So today I will share the company's knowledge about protein engineering of industrial enzymes. So this is today's outline. And first, let me introduce Amano Enzyme. Amano Enzyme is a Japanese-based enzyme manufacturing company. We have a history over 100 years, and we have experience over 70 years with enzyme business. This is our corporate slogan, specialty enzyme for every industry. We are producing industrial enzymes for food processing, pharmaceuticals, biotransformation, and diagnostic. I will explain about the history of Amano Enzyme. We founded in 1899, so that means we have over 120 years of history of business. So we started the production of enzyme in 1948. So in 1996, we decided to shift it to the specializing in enzymes. And 2000, our corporate name to change to Amano Enzyme. So this is our corporate slogan now, and our overseas sales ratio is approximately 7%. This slide shows the Amano's office over the world. We have an office in US and UK and China and Thailand. Also, we have a production site in China. This map is Japan. So our head office in the Nagoya city, and we have 3 plants, and 1 research center is located in Tokai area, the central part of Japan. So this is I'd like to talk about our social responsibility. So currently we are facing a sustainability challenge in food, energy, and environment. So we believe biotechnology is a cheap technology to solve them issues. So through the enzyme business, we believe we make a world a better place. So this is R&D section. So This picture is our R&D center as my workplace located in Gifu City. So our mission included these 5 topics: innovative new products, prototype development, and cutting technology, and commercialization, and product improvement. We have also R&D team in the United States. So US R&D team has extensive experiments and diversity. We support specialty enzyme solution to customer needs. Please contact us about industrial enzymes. Let's move to the second topic. This slide shows the example of plant-based food. Recently, many plant food products are launched, and such as alternative milk, cheese, and meats. In this presentation, I will talk about application of alternative milks. So, enzyme can modify the physiological and functional property of plant-based food. Therefore, enzyme can help make a more attractive plant-based products. So this is a plant unlimited products. We offer the different enzymes to meet customer needs. In this presentation, I will talk about protein glutaminase, PG. Protein glutaminase is a unique enzyme for food application. So our CTO, Dr. Shotaro Yamaguchi, isolated PG productive bacteria from soil in 1997. After that finding, we have commercialized as products. So that PG provided new value to protein integrants. We have been developing 30 years for enzyme for 30 years. So we published 70 papers and filed 88 patents. For using food processing, we must register each countries. We registered 43 countries already, and our 24 countries sales records. This is an enzyme reaction. So PG converting glutamine into glutaminate acid without a peptide chain cleavage. In this figure shows dissolving, unfolding protein higher structure and change to protein function. So therefore, PG make a protein integrants for more attractive, such as stability, mouthfeel, stability, and forming was improved. I showed one example of this effect. So this is an almond milk application. This is controlled, and this is PG treated almond milks. So PG prevent curdling in coffee. That controls are separated and curdled. But PG treated almond milk is not curdling in the coffee. So this application is popular all over the world. So next is move to the protein engineering. So we try to expansion of substrate function. So to overcome that limitation, we try to developing protein engineering. This shows our PG variant, thermotolerant PG, thermosensitive PG, and higher active PG, and broad substrate specificity of PG. In this presentation, I will focus on the thermotolerant PG. to develop thermostolant PG, we have tried to envelope the enzyme using a directed evolution. We made a total 3 round random mutagenesis and to thermotolerant protein glutaminase. Through the 3 round mutation, the reactivity is included 2.5 at the 55 degrees Celsius. Right figure shows the mutation point green colors, blue colors. So that's residues are located on the surface of protein, far from the catalytic residues. So we check the general characteristics of thermotolerant PG, named PG third. Optimal temperature was a slight improved to shifted to the 55 to 65. So thermostability is also improved as a shifted to the 60 to 70s. Optimal pH are not changed slightly, almost same as the original one. And pH stability is changed to the alkali tolerance. We also evaluated the specific activities. This is we check the 3 substrate. One is this is a synthetic substrate, and this 2 is a natural substrate. So as I mentioned, PG30 is shifted to the higher activity at substrate for synthetic substrate as same as a natural substrate. Specifically, milk casein. Using milk casein, the specific activities are improved dramatically increased. So we try to next application. Yeah, usually, plant-based protein have high starch content, so viscosity is important. So usually we try to the alpha amylase and reduce the viscosity like this. And after that, we react the PG. But alpha amylase need a higher temperature, so we try to the one-step reaction using this thermotolerant PG. So rice and oats both are high starch contents. So we try to the alpha amylase and PG put in the same time and react one reaction. This shows the result. So rice and oat milk production. Y-axis means a protein content. So in the case of using a PG third, soluble protein was included 1.6 for rice and 2.44 for oats. So our results, this result shows we can use a one-step reaction for higher starch contents integrants. This is conclusion. Thermotolerant PG has been developed, which are specialized for high temperature reactions. High starch content plant protein can solved by one-step reaction with a thermotolerant PG and alpha amylase. So we have a various lineup for the developing uses various plant-based proteins. This is our ongoing project. We are investigating treasure hunting by in silico approaches. In general, it is very difficult to find screening combination of mutation sites because that site is encode 20 amino acid. Therefore, ongoing project, we are trying to how effectively to found the combination. So we're using experimental data and machine learning approach. So we are trying to do some program to find a new variant effectively. This is a collaboration work, Nagoya University and Fujitsu Corp. In the future, I will talk about this project. So let's move to the third topic. Third topic is lipase. So third topic is a product of menthol. Menthol is a monotemperal alcohol, and this is a useful fragrant or some foods. This is a L-menthol. Menthol have a 3 catalytic center make and 8 isotopic optima. So L-menthol is a most preferable structure, and natural menthol, also L-menthol. So L-menthol is extracted from a plant such as peppermint. According to demand is growing, so chemical synthetic method is developed. So L-menthol is in the chemical method company, 3 major chemical companies synthesize the L-menthol on industrial scale. And also enzymatic synthesizers reported using lipase and esterase. This is the Barcolia cepacia lipase. So that is a commercialized enzyme PS Amano. Using a wild-type BCL, it is reported 79 enantiosen for L-menthol. We try to enhance the enantiosen selectivity because for large-scale production, high quality L-menthol is difficult to separate required, and this is difficult to separate D-L-menthol on the industrial scale. So we aim to improve the stereoselectivity of BCL and optical purity of L-menthol. In this study, we simulated the hydrolysis reaction of BCL using L-menthol methyl acetate and D-methyl acetate. And we have a MD simulation with L and D methyl acetate and BCL. We collaborated Japanese Research Institute, Institute of Advanced Industrial Science and Technology, AIST. So we expected molecular dynamic simulation. Using a molecular dynamic simulation, we expected short time developing on the modified enzyme. In this study, so molecular dynamic simulation using for the enhanced enantiosen selectivity. This is we had 3 steps of this process. First, prediction of unstable structures. The second, prediction of interaction between protein and substrate. The last part predicted enzyme design based on MSPR method developed by AIST. First, we try to reproduce docking poses of lipase and methyl acetate. So we found the initial structure was unstable. However, we found a stable cluster and lower energy. The structure seemed to be the closed form, so lipase usually have a lit structure, and the initial structure is an open structure, but stable structure looks like closed structure. So we speculate closed structures are more stable. This slide shows the way of enzyme design. A little bit complicated, but so I try to explain. Our collaborator, Dr. Ikebe and Dr. Kameda, developed the mutation site prediction for enhancing radioselectivity, MSPR method. So in this case, L-methyl acetate is a desirable substrate, and D-methyl acetate is undesirable substrate. We need to do MD simulation and docking simulation each substrate and check the amino acid interact the substrate. In this case, the amino acid A and B interact to the desirable substrate, so we didn't affect that enzyme amino acid. And desirable structure is a D-methyl acetate. So in this case, C and D is interacted to the D-methyl acetate. That we speculate that C and D is a candidate for the substitution. So we make a list, top 100 or something. So and ranking, make a ranking. So in this case, C and D is a higher ranking than A and B. According to that method, we apply the MSPR and make ranking list amino acid of residues. So we selected the top 3 residues for substitution candidate. This is show the graph Q88, G16, H15. That's located in the catalytic triad of BCL. So through the experimentation, we found some candidate. So we tried the stocking simulation and in vitro evaluations. So this is our result. That we found 5 variants, and especially these 4 variants over 99 purified. Interestingly, conversion rate is also improved compared to the wild type. That's for industry, that's an important point. So we think, sorry. 15 and glycine 16 lost their enzyme activities. So we think ability of MSPR can predict effective mutation point. So this is an acknowledgement. Protein glutamase project is our in-house project. So using with thanks to the Amano enzyme researchers. Lipase from Berkeley Sepacia is a collaboration work with AIST and Amano enzyme. And we had financial support from Japanese government. Okay, this is my last slide. So we Amano enzyme provide any enzyme solution to meet your need. Feel free to contact us. Thank you for attention.
any questions? Thank you. Are there any questions? I'll start with a question. So I had my oat milk this morning, and it was sweet, so I suspected there was an amylase that was used to make that starch a little bit sweeter. But I never thought that there would be a protease that also needed to be in there to stabilize it. So so the question is, soy milk are proteases needed also to stabilize the proteins in soy milk as well?
Yeah, our if you use a protein glutamase instead of protease, so maybe some reduce the bitterness or something because protein glutamase are not cleavage the peptide bond. That peptidase protease cut the peptide chains and make some amino acids.
Okay.
Maybe a little bit different. So
Okay.
these are application of protease applications.
Thank you. Are there other questions? I think it's on.
Thank you for the interesting talk. I'm Yuta Hayashi from the University of Tokyo, and my question is about the thermotolerant PG, and I would like to know the residue to introduce that mutation of the PG.
So
how did you choose that residues?
yeah, in this case, we just use random mutagenesis. So we didn't select any amino acid, just select the higher temperature activities.
Thank you. So some of the residues are very critical for that, and I want to know whether some of the variants are not produced as a soluble protein or and you.
So
sorry, I like to know that. How many variants did you try, and how many of them are very successful for that thermotolerance?
Yes, yeah, we tried third round, maybe one round 2 months, and totally 6 months or something. So repeated evaluations.
Thank you very much.
Are there other questions? Yeah, thank you.
Thank you for your presentation. So glutaminase. So you mentioned unfolded protein is active. So are there any some unfolding mechanism to activate the protein in cells?
Yeah, I think just to change to the protein surface. Yeah, if you hydrolysis some glutamine to glutamic acid, so we have a OH here and structure as relaxing and exposure inside of protein. So that means hydrophobic region is exposing. So that means we can see the protein properties are changed.
So, sorry. My question is, this protein in nature, so folded protein is inactive. And so to activate this protein, unfolding step is required.
Yes. That's a pro-mature type protein enzyme. So need a pro region and cleavage a pro region. So to move to the active form. So yes.
Okay.
So this protein glutaminase is toxic for the cell. So the pro form is inactive. And after the removal of pro region, the mature protein had a, and after the excretion from the microorganisms, then the active. So make sense?
Yes.
Okay. Yeah.
We do have one quick online question. It says, very nice talk. Which software have you used for MD? Thank you. Which software have you used for MD?
The software used for the molecular dynamics simulation. That was done by collaborators, so maybe you don't know.
Maybe I can see that in paper.
Okay.
Sorry, I didn't answer that.
Okay. I know that at the University of Minnesota, the Desmond is a popular molecular dynamics software that is available here.
There is one more online question. It says, great talk. After PG action, ammonia is forming. Does it change the taste of beverage?
Yeah, maybe not. We don't have not so big effect. Yeah, but exactly. So you said making ammonia. But usually in the food process, not so many big amount of ammonia, so does not affect strongly.
Thank you very much.
Thank you very much.